Biology and biochemistry. When you talk about living things on our Earth, there are 5 kingdoms. 50 years ago, when I was a child, we had 2 kingdoms, animals and plants. By the time I was in high school, I realized there's a third, genetically different kingdom, fungi, which included, the funguses, molds, mushrooms, all that. And then, in the last 30 years, we've also realized there are 2 additional kingdoms, prokaryotes, which is the bacteria, and the protoctista, which is basically the blue-green algae. It actually is a completely separate kingdom.
All right, we got 5 living kingdoms. 50 years ago, we thought there were 2, plants and animals. Then they realized there was a third one, fungus, which includes yeasts, molds, as well as mushrooms. So all kinds of microscopic funguses as well. In the last 25 years, we realized there are actually 5 kingdoms genetically. The protoctista and the prokaryotes, these are all separate kingdoms. The prokaryotes are actually blue-green algae. So there are 5 kingdoms total within our human living sphere.
These are drawings of common bacteria infecting humans, and you'll probably recognize quite a few of them. Staph infections, up in the upper right, you can see these right here. Right here, staph infections, often in hospitals. Salmonella, often from undercooked chicken right here. Anthrax, which has been used as a bioweapon. That's Bacillus anthracis. Tuberculosis, closely related to it. This is what causes a lot of pneumonia. Helicobacter pylori, which is actually what causes ulcers. I used to think it's called from worry and guilt, believe it or not. Botulism poisoning, Clostridium botulinum. Then E. coli, which we need in our intestine, but not in other parts of our body.
What you notice here are some of the common bacteria that affect human civilization. Top left, you got Staphylococcus. That is staph infections you often hear about in hospitals. Bacillus anthracis. Third one down, second from the right. From the left, excuse me. That's anthrax. Bacillus tuberculosis. That's tuberculosis. Helicobacter pylori. It's on the bottom left. That is what causes ulcers in stomachs for the most part. Clostridium botulinum on the bottom. That's called botulism, or canned food poisoning. Escherichia coli, E. coli, it lives in our intestine. Gets outside our intestine on food, it can be very dangerous to us though.
And these are viruses about 10,000, 100,000 times smaller than bacteria. They basically are protein coats surrounded by DNA. These are some of the more common viruses. The human adenovirus, which causes many colds right here. Poliovirus, which we do have a vaccine for. Oh, they all look like crystals. Herpes virus right here. There's even a measles virus right here and flu viruses. You can see, and those curly Q areas in the middle are actually the DNA.
Viruses. Just like the coronavirus affecting us all right now throughout our planet, viruses are about 1,000 times smaller than bacteria. There's even questions if they're living things or not. For the most part, as you can see in the measles virus, for example, most of these, in fact, are just a protein coat with DNA on the inside. You look at the cauliflower mosaic virus, and the human adenovirus, the herpes simplex virus, they look like crystals. The T2 bacteriophage just looks like some kind of weird satellite. These are viruses. As I said, we don't know if they are living or not. They're just DNA surrounded by protein.
Cytology is the study of living cells, whether they're nerve cells or blood cells, cancer cells. Cytology is the study of cells and all its components.
This gives you a quick view of how different cells look like. In fact, these are all cells in the human body. Red blood cells, Top right, it's a cancer cell on the bottom. And on the left is a nerve cell, which looks a lot like a flower.
Now, I'm not going to expect you to learn all the aspects of the animal cell and plant cell, but I wanted to point a few things out here. We've got the nucleus right here. Here's the nucleolus. The nucleus is right here. It's this big purple part in the center. And then the nucleolus within the nucleus, which holds their DNA. Right here we have ribosomes, where RNA comes to get the information of the DNA. The DNA is within the nucleolus. We also have the mitochondria, which are the powerhouses of the cells. They produce and digest the energy that the cell needs. The other difference between an animal and plant cell is an animal cell is protected because it's within the body of an animal. It's only got plasma membrane.
This is a model of an animal cell. As you see the nucleus, nuclear envelope, there are ribosomes, smooth endoplasmic reticulum, mitochondria. We're not going to go over all these. I want you to get a good view of what it is. The DNA, which we'll cover in a little bit, is within the nucleus. The mitochondria, where you see the name on top pointing to them, are the powerhouse of the cell where it derives its energy from. And then the ribosomes, which are below the nucleus, contain RNA. This is how information from DNA is captured and carried to other parts of the cell.
However, a plant cell right here has many similar things. They have the nucleolus and the nucleus. They have the mitochondria, as you can see here. But in addition to a cell membrane, which is on the inner part, they have a cell wall. This could be a leaf right here. And it needs protection from the elements. So in addition to a cell membrane, it has a cell wall.
Different than an animal cell, a plant cell also has a cell wall in addition to the cell membrane. It's protected from external elements. And on the inside, there's a vacuole. This is a large space of all their waste materials.
Now we'll talk about the carbon cycle in coming lectures. But right here, the carbon cycle is basically all living things are based on carbon. Our atmosphere is carbon dioxide. When we burn living things, or as they decompose, whether it's bodies of animals and plants, or even the process of burning coal and oil, this releases carbon dioxide into the atmosphere. Photosynthesis, where the sunlight hits the plants and the plants turn sunlight into sugar, also requires carbon dioxide as well. A lot of the carbon dioxide in our atmosphere gets absorbed into the oceans, where it's absorbed by phytoplankton and turns into sediments. They actually make bond to calcium carbonate with carbon dioxide, creating these shells. There are billions of microscopic organisms using shells, and that goes to the bottom of the ocean, creating our sediment, which turns into limestone.
The carbon cycle is basically part of our whole living process here. Carbon is within our atmosphere as carbon dioxide. There's carbon in the ocean as calcium carbonate or limestone. And then when we burn living things such as trees, wood, that's also carbon, and it returns back to the atmosphere.
The DNA, as I talked about, is within the nucleus, and this is the architecture of all living things. Everything that decides what's going to look like, how big it's going to be, the color hair, the color eyes in humans, the processes, how it digests, reproduction, everything is within this structure here. It is basically an architectural model for what all the cells should do in the entire body, in fact.
DNA is the blueprint of all living things. It has every component of what makes all living things look and act the way they do. DNA is considered a double helix structure, and really what it is, it's just a ladder that's twisted.
Now, as I said before, the DNA is located within the nucleus. And you see these chromosomes. Humans have 46 chromosomes, 23 pair. We'll go over that in a minute. But they look like X's. And really what those X's are, that is the DNA wound up very tightly so it can fit within the cell.
DNA, as I said earlier, is located within the nucleus of every cell. Within the nucleus, there are chromosomes. Human beings have 23 pair, which I'll go over in a moment. Each chromosome is that twisted double helix, twisted very tightly and is wound around. That, on the very right here, is the DNA ladder, as we're talking about.
Now, humans have 23 pairs, 46 chromosomes, and half come from the male, half come from the female during reproduction. There are several interesting aspects. We've just mapped the entire human DNA genome in the last 15 years. We still don't know a lot of it does. We just know the location of all the molecules for it. This is a male here because that XY. It's got a large X, the 23rd pair, and a Y. This is a female here. It's XX. Other areas we can look at here, there are also many defects that can happen if one of the DNA components is damaged. And when we look at this, we know certain chromosomes determine eye color, hair color, how tall someone's going to be, but we don't know where a lot of these are. There are some areas we know probably about 10 to 15%, but we don't know at 85-90% of what relegates how something or someone's going to look based on their DNA.
This is an actual picture of all the chromosomes within a human being in every cell. We have 23 pair, or 46 in total. Of the 23rd pair, where you see XX and XY, XX makes a female, XY makes a male. What we're looking at here are all the diagrams and the architecture of what makes a human being look like a human being. Variations in this can cause mutations. Down syndrome, formerly known as mental retardation, is an abnormality on the 21st chromosome. There are also sex-linked chromosomes. These are chromosomes that cause hereditary differences between men and women. It has nothing to do just with the sexuality of the person. For example, male pattern baldness is only carried by the female. It's only carried on the X chromosome. Other areas in the X chromosome is men having more facial hair than women, women being able to have children and men can't, and several other things, such as a man usually having a stronger jaw and more protruding forehead than a female. All these are on the X chromosome. The Y chromosome.
Now, DNA is almost a binary code. There are only 4 molecules that make up DNA. Adenine, guanine, cytosine, thymine. And they are in 2 rows. Guanine can only bond with cytosine. You can see guanine on the left, cytosine on the right. Here's cytosine on the right, guanine on the left. And then thymine can only bond with adenine. Thymine on the left here, guanine on the right. Adenine right here, thymine on the left. So if this thing gets ripped in half as it reproduces, it can only bond with its other matching pair. And really, like I said, this is called a double helix, but it's really just a twisted ladder. And this allows it, if there are rips or tears in it or malfunctions, it can still only bond with one other code. And it's basically ATCG, adenine, thymine is the AT, CG is cytosine, guanine.
The unique part about DNA, and this is with all DNA and all living things, from sea cucumbers to human beings to zebras to amoebas to parameciums to oak trees, there's only 2 base pairs. What I mean by that is there's 4 chemicals: adenine, guanine, cytosine, thymine. Guanine can only bond with cytosine. Adenine can only bond with thymine. As you can see in my diagram here, guanine is with cytosine, thymine is with adenine. The orders of these, and there's billions of them, determine what that individual component will look like. For example, it could be a series of guanine, thymine, adenine, cytosine, thymine, adenine that makes one person's eyes color blue, and maybe something slightly different, guanine, adenine, cytosine, thymine, makes another person's eyes brown. These are billions of base pairs long. But these 4 chemicals, only 2 bond at a time, allow it to split in half. And then each side is split in half. It can only bond with one thing. As I said, the guanine only bonds with cytosine. The thymine only bonds with the adenine. And that is how DNA replicates itself.
Now, in reproduction, we have mitosis and meiosis. Mitosis is basically the cloning of a cell. We take one cell and it splits into 2. The DNA basically goes down the middle and then splits in half and then doubles. So basically, you got a clone of it. And if there's a defect in one side, there's going to be a defect in the other. You're making a clone, and that's good or bad, depending on how you look at it. It's quick reproduction. But if the mother cell, which is what the original cell is called, has defects, then both the daughter cells will as well.
2 forms of reproduction. The one we often see about with microorganisms, single-cell creatures, is mitosis. Literally is a clone of itself. You got the 4 phases: prophase, metaphase, anaphase, telophase. And basically what happens is, if you look at this, is these are the chromosomes splitting in half. And once they've completely split apart, the cell separates into 2 pieces, and now we've got 2 new cells, each with the same DNA.
Now, to counter this, we have meiosis, which is sexual reproduction. All our cells within our bodies of all living things have 2 pairs of everything, with the exception of our reproductive cells, sperm in males, egg in females. Most cells are diploid, di meaning 2, and that means there are 2 pairs of chromosomes, except sperm has 1 pair. No pair, it's just a single 23, and then the egg has another 23. When those come together, we have 2 pairs. But see, both DNA is different, so as it comes together, some overpower the other, and that's why we have slightly different genetics. This actually counters the possibility of having defects being passed along. It greatly reduces it. Rather than have a clone with the defects are in the mother cell and both daughter cells. In this case, there are a great unlikelihood that when the haploids, that's what they're called, half of it, come together, they'd have the same amount of defects. This is why incest has been outlawed in almost all cultures, because they realize this. They didn't know the scientific rationale behind it. But when brothers and sisters marry, you saw like the ancient Egyptians or with the Habsburgs within Europe, if one side has a defect and the other side has a defect, the child's going to have a defect. Meiosis greatly reduces this as long as we have different families coming together.
Meiosis, or sexual reproduction, is what gives us variation. As with mitosis, as I said, when it splits in half, they're 2 of the same thing. In meiosis, we only have half the DNA, 1 from the egg, 1 from the sperm. And where we have 46 total chromosomes, 23 pair within our DNA, the egg only carries 23, the sperm only carries 23. So when they come together, that forms a new living creature. Whether it's an elephant, a human, a zebra, a flower. And that's why I get variation, is each one is slightly different than the other. This is called meiosis.
Interestingly enough, mitochondria, which is the powerhouse of the cell, has its own DNA, and they often do mitochondrial DNA for tracing back ancestry.
Interestingly, the mitochondria, the powerhouse of the cell, within a cell also has its own DNA. In fact, they can trace back mitochondrial DNA from mother to daughter. And they did this, and they basically figured out that we came from one family of women about 15,000 years ago in sub-Saharan Africa.
Now, as I said before, viruses reproduce by injecting themselves into a cell, into the nucleus, and taking over the DNA. And it says its DNA basically injects itself into the DNA of the cell and says, start making more like me, and keeps making viruses over and over and over until there's so many of them, the cell bursts, and all these viruses go out and start doing the same thing to other cells.
Viral reproduction, viruses. Another question if they're not even sure they're living beings, I should say. As I said before, it's literally a DNA packaged within a protein coat. The way it does work is that it injects itself into a cell. It goes into the nucleus and makes its DNA make copies of itself. It just starts manufacturing more and more viruses. Until there's so many viruses inside the cell dies and bursts. And what do these viruses do? They go to other cells and start telling those cells to make more and more of themselves.
Let's switch over a little bit to biochemistry here. Now, I'm not going to expect you to learn all this, but this is the periodic table of elements. I'm sure you've seen this before. We're talking a little bit about a carbon, and there's a rationale why carbon is so well studied in chemistry. Put it here, carbon is 6. And just a little background here. 6 is the atomic number. That means there's 6 protons. We'll go into that in a minute. And you can see here it's 12. That's the atomic weight. There's 6 protons and 6 neutrons. And this 6 is very important here, which we're going to cover in the next couple of slides.
This is the periodic table of elements. 103 elements within this. The heavier you get, the less there are. Hydrogen, number 1, the top left, makes up 99.9% of the universe. Helium makes 0.1% of the universe, and minuscule amounts of everything else is within our universe. Not a lot more on Earth here. And that brings up the debate of where we originated from. Probably a supernova, where they have most of these heavy elements. But I want you to look at a couple things here. I want you to look at number 6, where it says C, carbon. That is the base of all life, I said earlier. And because carbon is uniquely designed, is why carbon is within so many aspects of all living things.
All atoms have a nucleus, which is made of protons and neutrons in the middle. Protons have a positive charge, neutrons have no charge. And the reason they have neutrons is if two protons both have positive charge touch each other, they'll repel. The neutrons neutralize that. So the protons have a positive charge, and the electrons have a negative charge. And if we have 3 protons, we have 3 neutrons. We have 5 protons, we have 5 neutrons. We have the same number of electrons as we do protons.
Before we talk about carbon, I want to go over the basic atomic structure. All atoms have a nucleus, which is made of protons and neutrons. The proton being positive, the neutron allows the protons to be together, otherwise they repel. And electrons, which are orbiting the nucleus. These electrons are part of what makes carbon very important, which I'll show you in the next slide.
Now, what's important here is these electrons are put in certain shells, certain locations. And the first orbit, these are all orbitals actually, can only hold 2 electrons right here. There's the first orbit right here. It can hold 2. The second orbit can hold up to 8. The third orbit, you can see it right here on the very right. That's the first shell. First orbit has 2. Second shell has 8. The third shell has 18. Fourth shell has 32. Fifth shell has 50. Now keep in mind, if there's 10 protons, that means there's 10 electrons. 2 would go in the first shell. 8 would go in the second shell. But what if there's, like in carbon, 6 protons and 6 electrons? Well, first 2 will go in the first shell, and then 4 in the second shell. Now, it wants 8. It wants to put as many electrons as possible to maximize it. It feels more balanced. But with only 4 out of the 8 possible positions, it is looking for something to bond with it to feel more secure.
The number of electrons equal the number of protons inside the nucleus. In other words, carbon has 6 protons. That means it'll have 6 electrons, and they're equally balanced. Where these electrons sit are called shells. The first shell, the very innermost area, can only have 2 electrons. The second shell can have up to 8. So in other words, carbon, which has 6 electrons, has 2 in the first shell, 6 in the second shell. But it can hold up to 8 in the second shell, and that's important, which I'm going to go over in the next slide.
So, I've drawn a little carbon atom here, and it's got 6 protons and 6 neutrons, which means that 6 protons means that 6 electrons. The first 2 are in the first shell, the first orbital, then there's 4 in the second shell. But it wants 8, because 8 is the balancing point if it has on the second shell. So it's going to look for something to balance with it. And the outer shell is called the valence shell. And when we look at this, carbon has 6 electrons, 6 protons. It's got 4 valence electrons. That means there's 4 in the outermost shell. Got some more examples here. Hydrogen, which only has 1 electron, 1 proton. It'd like 2 because it wants 2 in that first shell. It has a valence of 1. Oxygen has 2 in the first shell. It has 6 in the second shell, which means it's got a valence of 2. It wants 2 to do it. Nitrogen would like to have 3 more. Carbon, as I said, would like to have 4 more. It has 4 in there, but it wants 4 more.
Here again is carbon. 6 protons, 6 neutrons, 6 electrons. And as I said before, the first shell can only hold 2. The second shell holds 4 for carbon, but it can hold 8. And see, it wants 8 to feel balanced. It feels unbalanced with only 4 when it can hold 8. And that allows it to bond with other things. In other words, hydrogen, which has 1, we could attach 3 more, 4 to that in total. Oxygen has 2 extra electrons, so it could share 2 of each.
One of the most common molecules in the universe, because of this, is methane, which comes from the decomposition of living things. It's got carbon in the middle. It's got 4 hydrogens. Carbon has 4 electrons in our shell. Each hydrogen has 1. But now, if they can share together, what happens here is the hydrogen now gets a second 1 electron, so it feels balanced right here. The carbon, it's sharing 4 in total here. And now it feels balanced because it's got 8. The more you've balanced the valence shell, the more stable it is. And that's what it's looking for. It's looking for stability. And this is one of those common ones, but there are other things that can bond with carbon. And almost all our molecules do this. There's a whole component of chemistry called organic chemistry. It really should be called carbon chemistry, because when they first started working on it, they realized that most living things had all these different carbon components.
Here's my carbon atom feeling balanced with 4 hydrogens. Carbon has 4 outer electrons. They're called valence electrons. Hydrogen only has 1, and it has only 1 shell, and it wants 2. So by 4 hydrogen working with 1 carbon, the carbon feels balanced because now it's sharing 4 more electrons. Each hydrogen is sharing an electron because it wants 2, and it is very stable. 4 hydrogens, 1 carbon, very stable element. This is methane.
Now, looking at the atomic table, these alkali metals, such as lithium, sodium, potassium, they're in the very left. They only have one in the outermost shell. They want to balance with something, and they're looking for anything. And
alkali metals, which are on the left of the periodic table, very explosive. In fact, if you look at these, very important part of our diets, but also in their pure form, very dangerous. Sodium and potassium, in fact, in their pure form, are kept in oil, because if there's any moisture at all, even moisture from the air, they're going to explode. So these liquids that you see can be kept in is oil.
They bond with something on close to the very right called the halogens, such as chlorine, bromine. When they come together, they're very explosive because they're so excited to see each other, literally. They are so unstable. When they come together, they become extremely stable, but that causes explosions in the process.
The halogens, such as chlorine, bromine, iodine, you all heard of these. These are chlorine in pools. Iodine for cuts. Bromide. These are also very explosive, especially when they run into what I was talking about previously, the alkali metals.
But what you get when you do this, you call it called the salts, sodium and chlorine. Sodium, being very reactive with chlorine, it causes, common table salt. And why do we need this in our diet? Well, because we need the hydrochloric acid, the chlorine, within our stomach for digestion.
Interestingly, when the alkali metals come together with the halogens, those last two slides, they form what's called the salts. Very important part of our diet. In fact, you look at something like table salt, which is made of sodium and chloride. We need the chlorine to make hydrochloric acid, which helps digest within our stomach.
Now, as I said, this is all carbon-based, and our amino acids, which is what makes up our DNA, four types of amino acids, and there's hundreds of amino acids, are these carbon-based compounds. When they come together, create a peptide. When all these peptides come together, it creates a protein. We often think a protein is meat, but there are thousands of proteins. And technically, a protein is a string of peptides put together. And a peptide is a group of amino acids together as a string. Now, when we look at DNA, deoxyribonucleic acid, that's in the nucleus, it has all this information. Like as I said, it's the architecture, but it also needs to get information out. And that is what the RNA does. Part of the DNA splits open temporarily, and it sends a message to a single strand called RNA. That RNA goes to other cells, other parts of the body. That's what those ribosomes are for. That RNA stands for ribonucleic acid. Now, the only difference between DNA, other than being a single strand rather than double strand as in DNA, they both have cytosine, they both have guanine, both have adenine. But instead of thymine in DNA, it has uracil. That uracil can bond with the adenine.
RNA and DNA. RNA is a single helix, where DNA is a double helix. When that DNA splits in half, it sends a messenger to the RNA, which has one side. RNA is almost like DNA chemically, except instead of thymine, look at the bottom right, it doesn't have it. It has uracil as a replacement. Part of the reason I bring up RNA is there are a new group of viruses, new in terms of we discovered them in the last 25 years, called retroviruses. They don't have DNA, they have RNA. And the problem with a single helix rather than double helix is it can change more quickly. Most famous retrovirus you all know about is the HIV virus, which causes AIDS. The reason we still don't have a vaccine is the DNA keeps changing. Vaccines are based on the DNA of the white blood cell recognizing it. Since in a retrovirus it has RNA, it can change much more quickly, which is even after 35 years, we still have not been able to develop a vaccine for HIV yet.